Hi students, we have completed the inheritance, what is called the classical genetics, under which we have studied the cytoplasmic inheritance in the last class. That lesson is over. We have to go for the molecular basis of inheritance in zoology, chapter 5. Now, in the case of cytoplasmic inheritance, there is one question. Name the organelles which are involved in or which are considered as inheritance vectors. Name the cytoplasmic organelles which are considered as inheritance vectors. Now the chloroplast and mitochondria. The chloroplast and mitochondria, these two are called inheritance vectors. Inheritance vectors, because they are responsible for the transmission of the character from the parent, that is from the mother, to the young ones. They are called as inheritance vectors. Now, we have to go for this lesson. So, the hereditary nature of an organism is defined by what we have the genome of an organism. The hereditary nature of an organism is defined by its genome. The hereditary nature of an organism is normally defined by its genome. It is a decider defined by its genome. So it's nothing but a long chain of nucleic acid molecules, and they provided information for constructing an organism. This is number one. So the hereditary nature of an organism is normally defined by its genome. It's a long chain of nucleic acid molecules that provided information for constructing an organism. Number two. So the genome, you know that one complete set of hereditary information. If you are taking genome, a complete set of hereditary information. Complete set of hereditary information. For any organism, it's a complete set of hereditary information for any organism. So normally the genome is responsible or needed for constructing an organism. Again, if you are taking a genome in a diploid set of organisms, it is a complete set of hereditary information of any organism. Now the genome is divided into a number of different nucleic acid molecules. So it is being divided into number of nucleic acid molecules. The genome is divided into number of nucleic acid molecules, and each nucleic acid molecule has a large number of genes. So each molecule has large number of genes. Now what is a gene? The last one, regarding this one, what is a gene? Gene is a sequence within a DNA that represents a single protein. Gene is a sequence. Gene is a sequence within a DNA molecule. Within a DNA molecule that represent or that is responsible for, or that represents, we can say, that represents a protein. That represents a protein. That means it has a code for the expression of one protein. That's a meaning. We will talk about later under the transcription process and translation process. What is a gene? Gene is a sequence. Actually, within a DNA molecule, that means we can say a segment of DNA molecule that is normally represents that normally represents what is called a protein. So this is all the introductory part. So what is a genome? Now it is a material that is needed for constructing a living organism. It contains information, a complete set of information, and it is divided into a number of what is called nucleic acid molecules. If you are taking each sequence of nucleic acid molecules, it has a large number of genes, and each gene is a sequence that is within a DNA molecule, and that represents what is called a protein. This is a definition of gene. Now in this lesson, we are going to study the following chapters or the following informations. We are going to study number 1, DNA structure. Number 2, the replication of DNA to produce copies of DNA. Then the third process, the transcription, the process of making of RNA from DNA. That process called transcription. That is nothing but copying of mRNA and DNA. We'll talk about. Now the next process, translation. For every just amino acid there is a code. There is a genetic code. That genetic code normally determines a sequence of that is amino acids in a protein molecule, and that process called translation. Then how far the various genes are regulated. One gene is regulated by another gene. How far the gene is working, and that is gene regulation. We have to go for lab, operon model, etc. Then finally, what is the importance of genome sequencing? That is nothing but what are the nucleotides present in our body, in the nucleic acid in a correct sequence. That is called genome sequencing. Sequencing a basis in a nucleic acid. That is called genome sequencing. What is its importance? These are all the lessons we have to talk about just actually in this chapter. Now, we start with the genes, what we call the functional unit of inheritance. Now let us talk about the genes, the functional unit of inheritance. Something about the properties, some of the properties of genes. Now, gene is a physical and functional unit of heredity. Physical and functional unit of heredity. We already studied. Now the concept of gene was first provided by Mendel in 1860s. But he never used the term that is what is called the gene. He didn't give the name gene. He used only some factors in the chromosome responsible for the transmission of characters. He used the term only fact. The term gene was provided or introduced by a Danish biologist by name Wilhelm Johansen in 1909. Wilhelm Johansen in 1909 was the first person to use the term gene. He defined the gene as genes are discrete determinants of inherited characteristics. So he defined a gene as discrete determinants, that is the particles, determining the characteristics. So discrete determinants of inherited characteristics. That was the definition given by Johansen for the gene. Now we have to go for the next one, what is called the classical concept of a gene. There is another one classical concept of gene provided by, that is Walter Sutton. We will discuss this one now. I want to explain the classical concept of gene. This concept was provided and introduced by Walter Sutton in 1902. So he only just proposed the chromosomal basis of inheritance. Sutton and Boveri only proposed the chromosomal basis of inheritance. We have the lesson in botany, let us discuss later. And he was the one introduced what is called the classical concept of gene in 1902. What he said? According to this concept as proposed by him, number 1, the genes are discrete units, particulate units. Then, number 2, they follow Mendelian rules of inheritance. One gene, one character. That is the Mendelian rules of inheritance. A single gene expresses a particular trait. The third one, they are located on a chromosome in a definite locus. That is, a position of a gene on a chromosome is called a locus. For first locus, second locus, third locus, like that we are using. We are not using the term point. So they are placed or located in definite locus on a chromosome. And finally, they are responsible for the expression of the phenotypic characters. That is the Mendelian characters. The expression of the phenotypic character is because of, see, the inheritance unit, the functional unit, that is a physical basis of heredity, that is a gene. The number of genes in an individual cell is more than the number of chromosomes. That means in each chromosome we have several genes are present. For example, in the case of golden pig plant, we have in the fourth chromosome there are 3 genes. In some chromosomes we have 1 gene, but we have several genes are present in each chromosome. So the number of genes is always higher than that of the number of chromosomes. This is one of the additional characteristics. These are some of the main characteristics we can say under the classical concept of gene. Now they are arranged in a linear order like this, just like the beads in a string. They are arranged in a linear order, just like the beads in a string. The order will not be changed unless there is a mutation. And again, I mentioned already, they are located on a specific point in a chromosome, what is called as a locus. Now this is the locus. The point where a particular chromosome, so a particular gene is located on a chromosome. Now each gene shows many alternative forms, that is called alleles. Suppose you are taking what's called T gene, as in already marked. Or if you are taking capital A, we have alleles like this. So each gene has a number of alleles, number of alternative forms. These alternative forms are called alleles. Now the gene is capable of undergoing sudden change in its structure and composition. Composition means nothing but a change in the chemical base, the bases, adenine, guanine, etc. So change in what is called just the structure of the chromosome or structure of the gene or the position. That is called mutation. Such sudden changes in a gene is called gene mutation or point mutation because it is normally affecting a particular locus, hence called point mutation. Then also, as the DNA has a property of undergoing replication, capable of producing its own copies, that is replica, and that is nothing but the replication of gene. So each gene is also capable of undergoing replication, producing its own copies. These are all some of the properties of genes and the classical concept of genes. Also the question may be asked, what are the important properties of genes? How to pass on to a heading in search of genetic material. What's the meaning for that one? We have different components in the cell, RNA, proteins, DNA. Either RNA or proteins or DNA is a genetic material. That is in search of the original genetic material, either DNA or RNA, protein, through experimental analysis. Now, the chromosome was discovered in 1848 by a German botanist, Wilhelm Hofmeister. When he was observing mitosis, he just observed small rod-like structures. Then he used the term not chromosome. The term chromosome was given by wall layer. So he observed these rod-like particles. He didn't give the name, but the name was given by wall layer. So the discovery came only from Wilhelm Hofmeister, a German botanist, about the chromosome. Then it was Friedrich Meister, another person. In 1869, a Swiss physician, he isolated nucleic acids. He isolated a substance from the nucleus. He didn't know it was nucleic acid. He isolated a substance from the nucleus and called this substance as nuclein. But he didn't give the name nucleic acid. But the name nucleic acid was given by Altman in 1889. The name was given by Altman in 1889, the term nucleic acids. And it was known in 1920. All the chromosomes contain DNA or simply the nucleic acids and protein. That came in 1920. Then the transformation experiment conducted by Griffith, what is studied in your 11th standard, proved DNA is a genetic material. But he didn't know whether it was actually the transforming principle was DNA or not. So there was an experiment what's called the bacterial transformation. A non-virulent, that is bacteria, gets converted into a virulent form, a toxic one. We'll discuss later. And that is called bacterial transformation in the case of E. coli bacterium. That provided the first proof that DNA is a genetic material. The transformation experiment done by Griffith and followed by 3 other scientists. We'll discuss. Then that one actually gave the first proof DNA is a genetic material. But Griffith did not know the cause of transformation. One type of bacteria, what is called R strain, in his experiment, get converted to S strain. We'll talk about soon. This is non-virulent, non-toxic, and this is virulent, causes the death of mouse. That's why it's called as, that is virulent strain. This is non-virulent strain. So in his experiment, the R strain gets transformed into S strain. He didn't know the cause of transformation. Which substance is transferred from that is S to R, leading to the development of R strain. He didn't know. And also, the biochemical nature of the genetic material was also not known by him. So later, just in 1944, only 3 groups of scientists, they have just redid the Griffith's experiment in order to find out the transforming principle. The one responsible for converting one strain, that is R strain, into S strain. That is a separate experiment we have later. So this is about what we have in search of genetic material. What are the historical backbone that came, that is from people, to know about the DNA. That is what we have, a part making the gene. What is one gene, one enzyme hypothesis? This concept was given by George Beadle and Edward Tatton. They conducted an experiment in the case of one red mold, Neurospora crus. Now, before entering that is the example, and here, for every actually formation of a product, we need a biosynthetic pathway. A biosynthetic pathway involves, now this is a biosynthetic pathway, I represented S1 to S5. Each biosynthetic pathway involves a stepwise conversion of one substance into another. Here S1 is the raw material substance and S5 is the final product, in between which we have a number of intermediate products. So this is what's called biosynthetic pathway. A number of steps are involved to produce a particular product from the raw material. Now, each step, S1 to S2, is catalyzed by an enzyme E1. Likewise, S2, S3 by E2, E3, E4. These are all the enzymes for catalyzing a particular step. And each enzyme production is under the control of a specific gene. For example, E1 is being produced under the control of gene 1. That gene is responsible for the production of enzyme 1. That catalyzes a reaction, that is S1 to S2. Likewise, the gene G2 is responsible for the production of E2. So its production is under the control of G2. So the S2 gets converted to S3. Likewise, each step is catalyzed. This is called one gene, one enzyme hypothesis. A single gene controls the production of a particular enzyme. Sometimes what will happen, the gene G2 undergoes mutation. A mutation occurs in gene G2. So there is no production of this E2 enzyme or a defective enzyme will be produced. So that there is no conversion of S2 to S3. That results in the accumulation of S2 leading to the genetic disorder. So for example, phenylketonuria. Now we have the phenylalanine. One amino acid gets converted to tyrosine by the enzyme phenylalanine hydroxylase. Phenylalanine hydroxylase. This is in the liver. It is under the control of production of one gene G1. If this gene gets mutated, then what will happen? There is no production of phenylalanine hydroxylase enzyme in the liver. So there is no formation of tyrosine from phenylalanine. The phenylalanine gets converted to phenylpyruvic acid that is excreted in the urine. That results in PKU, what is called phenylketonuria disease. So many digestive disorders are because of inborn error of metabolism. What I represented is nothing but inborn error of metabolism. So there is inborn error that is right from the birth because of the defective gene. So that there is no specific enzyme, there is no conversion of a particular substance into another, leading to accumulation. We have the disease. Now in the case of just red mold, what he did, he just induced mutation in the case of the spores of neurospore. The spores are allowed to grow in a medium, but they are unable to convert one substance to another because of mutant gene. So what is happening? They are mutant variety. Such varieties need the addition of what is called one substance, one intermediate product that is missing. Then only the biosynthetic pathway is proceeding in the case of that organism. And such varieties, they are called the mutant varieties. They are called nutritional mutants because unable to convert one substance to another, one product in between the two. For example, between S2 to S3 or S2 to S4, this substance is missing in the case of mutant variety of neurospora. So it needs the addition of S3, then only the process is actually continuing. Now such mutant varieties which need the addition of what is called mutants to complete the biosynthetic pathway are called auxotropes. Auxotropes. These are called nutritional mutants. Nutritional mutants. This is also an ending question. Nutritional mutants. So these nutritional mutants are unable to convert one substance to another because of the absence of that particular substance due to mutation in a particular gene. So that what is happening? They need the addition of such missing substance. Such nutritional mutant varieties are called auxotropes. This is one. So this is a concept they have made. Each gene is responsible for the production of just one enzyme which catalyzes a particular step. But later, this concept of one gene, one enzyme was replaced by one gene, one polypeptide. One gene, one polypeptide. Hypothesis. Here, as the enzyme is made up of many just actually polypeptides. It's not made up of only one polypeptide, made up of many polypeptides. So they have modified the concept. One gene controls the production of only one polypeptide and not an enzyme. So this is the modified conclusion of one gene, one enzyme concept. Here one gene produces only one polypeptide of that enzyme. So one enzyme production is under the control of many genes. So this is called one gene, one polypeptide hypothesis. This concept was proposed by Enzyme and et al. Co-workers. Enzyme et al. Proposed a concept what is called one gene, one polypeptide. They modified the concept of one gene, one enzyme hypothesis to one polypeptide. One enzyme is modified, one polypeptide. So one gene controls only one polypeptide because the enzyme is made up of a number of polypeptide chains, not only one. So this is a new concept, what is called one gene, one polypeptide, proposed by Enzyme and et al.